This is Dr. Launen from Keene State College, and this is the second part of a lecture series addressing the sort of overviewing transcription. We're really going to focus in on, introduce you to the 3 stages, and then focus in on the initiation of transcription. So there are 3 basic stages to transcription. They are initiation, elongation, and termination. And what that really means is initiate is to begin transcription, and it's all about getting the RNA polymerase attached to the gene at the correct position. Elongation means synthesize RNA, so it's really all about forming phosphodiester linkages between RNA nucleotides and ensuring that the correct RNA nucleotide is in the growing RNA transcript. And the third and final stage is termination, or stopping or halting transcription, and that is mediated by particular sequences that are written into or encoded in the end of genes. So remember that transcription means that RNA polymerase is going to utilize the template DNA traveling 3' to 5' as it builds RNA 5' to 3'. That orientation is important. So the initiation stage involves promoter recognition, which means that RNA polymerase is going to recognize a sequence called the promoter at the front or 3' side of the gene, and it's going to attach. Chain initiation means that the RNA polymerase is going to ditch its sigma factor and become a core enzyme. So back in 1 it was a holoenzyme, in 2 it's just going to be the core enzyme, and then RNA polymerase is going to travel down the gene, opening it up and reading it and making RNA. And then in termination, we're going to see again the role of some specific sequences that halt transcription, and in some cases some additional protein factors. So let's focus on initiation first, which is actually the most complex part of transcription. And ultimately, it's really important that you know that initiation of transcription is all about promoters and how they interact with protein. The protein here is going to be RNA polymerase, especially its subunit called sigma factor. And we're really going to focus in first on the prokaryotic system. We're going to focus a little more on that, and I'll add in a little detail about eukaryotes as we go along. So a promoter is a sequence of DNA on the 3' side of the gene that binds sigma factor of RNA polymerase. That's a definition that applies to bacteria. If I was to define a promoter in eukaryotes, I would say it's a sequence of DNA on the 3' side of a gene that binds transcription factors to initiate transcription. So promoters are DNA sequences. So here I'm circling a very common structure, a very common pattern that we see in almost all bacterial promoters, and they look like this. They are palindromic sequences, so they're mirror images from left to right around a central point, and they're rich in T's and A's, and they are located very specifically on the 3' side of the start site for transcription. So +1 is the transcription start site. It's the place where transcription will begin. In other words, it's the place where the first RNA nucleotide will be formed. Upstream of that, and we're using negative numbers to show you that it's upstream, is a sequence called the Pribnow box in the -35 area. Both of these are consensus sequences. Consensus means to have agreement or to be the same. And in this context, what that's saying is these sequences are common in all bacterial genes, or essentially all bacterial genes. So they look like this in the Pribnow box, and they look like this at the -35 site. The promoter is located here. Its job is to bind the protein subunit of RNA polymerase called sigma factor. Once sigma factor is attached, then the rest of the RNA polymerase will attach, and it will create a transcription bubble, which means it will break the base pairs that are holding together double-stranded DNA in the gene coding area, and transcription will begin at the transcription start site. And then it will continue all the way down the coding sequence until you get to the area called the termination region. Okay, so I want to make another point here, and it's the use of the phrase UTR, or untranslated region, which you've been introduced previously in class already, but I want to remind you about that. So when the RNA transcript is made using all of this sequence, okay, and again, the sequence that will actually be used is the sequence that's shown in like this pinkish, peach color here, because the gene will be used 3' to 5'. So the coding sequence, like the template sequence is actually down here, right here, okay? And so when that sequence is used to make a messenger RNA molecule, it's going to have ends on it that are not actually coding. So the green area in here is the coding. That's a sequence that's going to result in amino acids, okay? Here's a start codon, and here's a stop codon, not really drawn to scale. And then here's some extra sequence here and here called UTRs, 5' UTR and 3' UTR, okay? Because it's complementary to the template strand. So if this ends 5', this end has to be 3'. Then if this ends 3', this has to be 5'. The untranslated regions are, think of them as like handles on the ends of the messenger RNA. They promote stability, and they also promote the initiation and the termination of translation. So they're really important, but they're not actually coding. That's a very typical looking structure for a bacterial gene. Eukaryotic promoters are more complex. So in a eukaryote, you have introns, or non-coding intervening sequence, or sequence that's in between the coding sequence. Coding sequence is encompassed in structures that are called exons. You can have literally hundreds of exons or dozens of exons within any given eukaryotic gene. Eukaryotic genes can be much larger than prokaryotic genes. The exons that are on the very ends, which in this case will be exon 1, and then here it's exon 3, but if this was a bigger gene, it could be like exon 35 or whatever, like the last exon. They are going to actually include the UTR regions that I just mentioned. So they won't be all coding. These will be all coding, anything in the middle. So we've got our introns and exons, and all of that will be used to make a messenger RNA molecule, okay? And then that messenger RNA molecule will be processed to remove intronic RNA. Later on, we'll talk about that in a moment. Common feature to the previous slide where we looked at a prokaryotic gene is the +1 site. So that is that's the location where you're going to use the first template DNA nucleotide to make an RNA nucleotide. We've got 5' and 3' UTRs, okay? And in a eukaryotic gene, the UTR also holds the poly A site. And we've got upstream of there, we've got a structure called a promoter. That is going to bind not sigma factor, because there is no sigma factor in eukaryotic RNA polymerase. Instead, it's going to bind a set of different proteins that are called transcription factors that work with RNA polymerase in eukaryotic systems. They're different, they're not subunits, but they're like helper separate proteins, okay? And many eukaryotic promoters have what's called a TATA box, but not all. So there's not quite as much consensus in promoter sequences in eukaryotic genes as we see in prokaryotic systems. Another feature that we see a lot of in eukaryotic genes are what are called proximal control elements, okay? And so these are like additional sequences that can bind regulatory proteins that influence whether transcription happens or not and the rate at which transcription occurs. Some additional sort of terminology here. Proximal promoter elements, these are the ones that I mentioned here, and they're typically within 200 nucleotides of the transcription start site. And then distal promoter elements, aren't shown here. They can be like thousands of base pairs away, or even on a different chromosome, and they are regulatory sequences that will bind, helper proteins that will play a role in turning transcription up or down. So let's go back to bacteria for a minute and talk about initiation. So here is our gene. Here's the promoter sequence in the gene. The yellow is sigma factor of RNA polymerase, which is a subunit of bacterial RNA polymerases. The entire purpose of sigma factor is to bind to the promoter region of a gene, and this little symbol means sigma. Okay? Once that sigma factor binds, then the entire core enzyme binds, forming what is called a holoenzyme, the entire intact RNA polymerase, which is able to promote the opening or breaking of base pairs in the double-stranded DNA molecule, creating a structure called a transcription bubble, which gives RNA polymerase access to the template so that transcription can begin. It will begin at the +1 site. And that's really all there is to it. A moment later, after RNA polymerase has initiated transcription, that sigma factor is going to come off. It's going to remove itself from this structure, and it's going to go off and find another core enzyme and repeat the process. In eukaryotes, the initiation is a little more complex. Actually, it's a lot more complex. But there are also some common factors. So we've got our gene sequence, and here's our template strand, and we've got a promoter sequence, and we've got the transcription start site, just to reorient you on this slightly different looking figure. What's happening first is transcription factors, which are proteins that are separate from RNA polymerase, but they work with RNA polymerase. So they're not considered the same protein, they're their own unique protein. Those transcription factors bind to the promoter region, and then they recruit in or attract the whole RNA polymerase enzyme. And this is showing me RNA polymerase 2. Remember, there are 3 different RNA polymerases that exist in eukaryotic cells. RNA polymerase 2 is always responsible for transcribing protein coding genes, so this gene must be a protein coding gene. And at this moment in time, once the RNA polymerase is attached at the promoter region with the help of transcription factors, the transcription bubble forms. You have to break the base pairs connecting double-stranded DNA in order to do that. And then here's the new RNA transcript being formed in the transcription bubble in the 5 to 3 direction, therefore using the template in the 3 to 5 direction. What's going to happen to these transcription factors? They are going to dissociate or come off from the promoter region and leave the holoenzyme, leave this core enzyme, to continue on in the elongation stage. And that's what we'll talk about next.